Started.
Okay,
So
great.
hello, everyone. My name is Lisa Sawsville. I'm the Executive Director of Vermont Coverts Woodlands for Wildlife. We're a landowner organization here in Vermont that works to help improve forest stewardship and wildlife management through a peer-to-peer network. And I'm excited to see many of our cooperators on board. And if you're not a cooperator and you have questions, you can type those in the chat box and we'll answer those, or you can email me at lisa@vtcoverts.org. You can also sign up for our e-newsletter on our website, and I'll put those information in the chat box. Tonight, we're excited to bring you the past Executive Director of Vermont Coverts, Farley Brown, who is a faculty member in ecology at the Sterling College up in Craftsbury. And she's going to talk to us about watershed bioassessment tonight. So Farley, I'm going to turn it over to you and let you tell us a little bit about yourself if you like, and jump right into the presentation.
Okay, sounds perfect. And everybody can hear me okay? Yeah, okay, great, good. Well, yeah, good evening, everyone. This is so exciting to be back with Vermont Coverts. It's been a while since I've been involved and had a chance to, I've heard of people's names and I definitely keep in touch by looking at the newsletter and online. But yeah, I'm so glad to be with folks today. And I also see a lot of folks from my sister-in-law down in Brooklyn, hi, all the way to my neighbor in Craftsbury, and then again, some of the Sterling students as well. So as Lisa said, I'm in the college program of, the Sterling College Ecology program, and where we teach a variety of courses, everything from natural history of the North Woods, vertebrate natural history, wildlife conservation. I also teach an environmental law class and land use planning. I really kind of go across the spectrum. We have students who are they give us such inspiration to come to work every day and to be out in the field. They ask great questions all the time and really just make it just a great experience to be teaching. So I'm also going to have a student come into the talk tonight, Connor Quinn, who is a student who's in his senior year, and he's done a lot with watershed assessment as well. I'm going to go ahead and get started, and I will not be looking at the chat myself, but if anyone has a question and folks want to put it into the chat, if you could just let me know. This is definitely something where I'm willing to stop and to take questions, or we can wait till the end, whichever way. I'm really excited to talk about the Black River watershed. This is where I've lived for over 30 years, and it's my backyard. And Sterling College is situated up in Craftsbury Common, but the Black River, which is about 26 miles long, starts off really in Craftsbury, and then drains all the way up in Newport in Lake Memphremagog. So the Black River watershed is part of the Lake Memphremagog watershed, and our watershed drains just about 134 square miles. So tonight I'm going to talk about aquatic habitat throughout the Black River, but I'm also going to be focusing a lot on the bioassessment, the research that the students have been doing with me for over 10 years now in the upper ends of the Black River, specifically in Craftsbury. So I always start off my classes, what is a watershed? Well, folks might know, or we all have a sense of what it is. It's really a catchment basin. It's a place where the water hits the height of land and drains into one central point, and then drains on out to the lowest part. So it is an ecosystem of its own. The watershed is, but it's made up of a variety of different types of ecosystems. And so they're draining through these natural communities, as well as going through a lot of the landscape that we've been changing through our land uses. So yeah. In Coverts, we talk a lot about connectivity of habitat and really how do we protect or how do we conserve or steward the large landscape to provide those habitat needs for a variety of species. And so we have the megafauna, we have moose up here in the Northeast Kingdom, we have all the different birds like the kingfisher. tonight we're going to talk a little bit more about those insects, mostly the larvae that are in the rivers and the streams that are there for all the way up to about 2 or 3 years before they hatch and live a very short life on the landscape. These bugs tell us a story. They tell us a story of the health of the water, and the river itself is a reflection of the health of the landscape too. So there's that interconnection, that tie between the terrestrial and the aquatic. And again, tonight we'll be focusing in on the aquatic. Again, here's the Black River Watershed. It's the Black River Watershed is just one of 3 different watersheds, actually 4, that drain into Lake Memphremagog. Lake Memphremagog, only about a third of it is in Vermont and in the US, and the rest is up in Canada. So what happens here in our landscape definitely goes right above along the lake up into another country. So we need to be very careful about what we're doing on the land, and if we're adding more and more, say, phosphorus, that's not just going to go out. It's going to go off our landscape, but it's going to also end up in Canada and then their drinking water. So at Sterling, we've been teaching the watershed analysis course for quite some time, probably at least 20, 25 years. And what I've been doing with the course since I started teaching it about 15 years ago is that I've been working with students to really focus in on analyzing not just the watershed, but the water quality of our water bodies, mostly rivers and streams. And we do this assessment in a variety of ways. We look at the biological indicators, the physical attributes, as well as the chemical indicators as well. We at the college spend a lot of time looking at the benthic macroinvertebrates, which we'll go into a little bit more depth, but we also do spend a lot of time looking at the physical, really better understanding that fluvial geomorphology, which is one of my favorite ways of saying the physical landscape of the river. We do at the college also do look at some really preliminary, we do a preliminary assessment of some of the chemicals like pH, dissolved oxygen, phosphorus, and nitrogen, but we don't get into that in great detail. But when we do an analysis, we're also looking at those natural communities, both the flora and the fauna, and we're looking at the land use history at the past and the present. And I always like to start with the historical land use. But before I get into that, I do want to say that the protocol that we use at Sterling is a protocol that's used around the world. It's inexpensive, it's easy, and we've been teaching it in a variety of different places. My students and I traveled to Central America, to Belize, and we actually share this bioassessment protocol with a lot of the school groups. And here we're in Golden Stream, which is a small Mayan community, a Mayan village. And so the kids were just so thrilled to be able to look at the macroinvertebrates while their moms are upstream washing the clothes and washing dishes. And so now the mothers were getting all concerned that there were hellgrammites and all these other scary bugs in there too, but they're all bugs that they serve a purpose. So focusing up here in the Black River watershed, we're in the Northeast Kingdom. A lot of people, when we think about historical land use, we recognize that the land was open, say 80% open for the most part throughout New England, and 20% forested. Well, a lot of it had to do with agriculture. Up here in the kingdom, it was predominantly logging. Logging was a major source of the economy here, and a lot of the wood resources were used locally, but then also were shipped out of town. This is Craftsbury, and for those of us who live in Craftsbury, you might recognize this as Norman Turner's house. The landscape up here going along Calderwood Road, completely open. And again, a lot of the wood was feeding this beautiful old sawmill. The sawmill was, it's really fascinating sawmill in a variety of ways, but in part because of the granite that they used to build up the foundation. For those of us in Craftsbury, we recognize this bullseye granite, which is a very unique granitic form of that's only found in a couple places in the world. And so we're pretty proud of it up here in Craftsbury. Bullseye granite, it's an avicular granite, and there are other people on this Zoom who could tell you a lot more about the geology than I can. So we look at this historical landscape. I've gone back to where this mill was once sited, and I could just about find only the retaining wall that was left over from the mill. And so I understand by looking at the vegetation now, I quite often look at the landscape and think, what did it look like historically when it was all open? What did the sediment, how was the substrate of the river? And I also often wonder, what were the bugs, the macroinvertebrates that were living there? The Black River continues to wind up north. It actually is a fascinating river because it does flow south, kind of south through Craftsbury Village, then takes a big U-turn and starts flowing north to Lake Memphremagog. So this is the Black River meandering along the valley through Albany on up to Irisburg. These are the Lone Mountains in the background. And again, you just see that the landscape is over, no real riparian buffer whatsoever. Up in Irisburg, there was a major sawmill up there. A lot of the wood was actually brought into the yards up here in Irisburg, or up there in Irisburg, and the logs were then floated or used at the mills, but then also floated up to Newport. South Bay, which is just outside of Newport, was where a lot of the wood sat. And if you go there today, my understanding from some people is that you can find remnants of some of the wood that was actually stuck in the bottom of South Bay. Of course, this open landscape, what it did was that it provided an opportunity, not opportunity, but the water ran off the landscape and created these major floods. So the 1927 flood, along with the 38 storm, really devastated a lot of the communities, especially up here where the landscape was washed off. The rivers really changed their beds because of these major flood events, and we see that happening even more recently as well. I'm just going to show quickly, this is one more mill up in Coventry, Vermont. And when you think about the mills, they had to dam up the river in order to get the power to run. So the damming up of the river would build up all the sediment behind the dam, and once again, change the substrate, change the landscape. We go back there now. This picture was actually taken in 2000. And we can still see a retaining wall that was there from that mill, but how the landscape has grown back in, the willows have come in, the alder have come in, silver maple looks like in the background, there's some pines, but so the river is reestablishing itself and the natural communities are flourishing again. This is now what we have to be concerned about, is our immediate land use. So we look at the history, we know the river responds and it can get back into shape, but the more and more that we add any type of sedimentation or other pollutants into the land, then we're going to start seeing some major impacts to the health of the ecosystem, the biodiversity of the riverine systems. In Craftsbury, down by off what we call the Cemetery Road, it was a big farming area. Now we are down to just one smaller farm. And it used to be, before rules and regulations and incentives came in place, it was that the cows would actually go into the river. Nowadays, the cows are fenced out of the river. But this is back in 2006, I believe, when I was out there with some students and just seeing the stream bank failure that was happening in that area. There was a study done back in 2011 by Northwood Stewardship Center. They were actually contracted with the Lake Memphremagog Watershed Association to put out, produce testing for phosphorus and nitrogen. So here we are in Craftsbury, right here at the headwaters, and then we get out here to Lake Memphremagog, to the north end or to the end of the Black River watershed. But upstream here, we had some farms that were really high in phosphorus. Since then, several of the farms have gone out, and there also have been practices put into place to help mitigate the runoff of phosphorus from the farms. We also have to think about a lot about our stormwater infrastructure. And in Craftsbury Village, we have a very small stormwater infrastructure that is all drains one pipe ultimately into the Black River in the village. We see that there's also some of the land uses have really been forcing the river to become narrow or to encroaching upon the river's floodplain. And there's been the river's response is to move back and forth against those stream banks. And we have a lot of stream bank failure happening around our town garage, off the creek road, where the rivers also are being, the road is impacting the floodplain. We're seeing a lot of that stream bank failure. I do have to say that there's part of the stream bank failure is definitely a natural cause with the different, the seasonality of our area. So big snow melts and rain events will naturally, the river will move and actually begin to erode away at the stream banks. But because of our land uses, we have to be careful not to create even more of a problem. And the problems ultimately, again, come in different forms in terms of impacts to the river health. And I always stress to the students that sedimentation is one of the major pollutants up here in this part of Vermont, since we don't have a lot of urban development. But so sedimentation is an issue here. We see a lot of sedimentation in the village, Craftsbury Village site, in part because of the soil that's getting into the stream banks, or into the river because of the stream banks failing. So sedimentation definitely becomes an issue, especially for those of us who are thinking about wildlife all the time. It's creating a major problem with the wildlife habitat, specifically for those macroinvertebrates, as well as for fish. So macroinvertebrates. These benthic macroinvertebrates, they're benthic, they live at the bottom of the river. They are indicator species. Each particular animal has a different level of sensitivity that, basically an ability to adjust to certain modifications of the river because of either land use changes or storm events. But some are so sensitive that they can't handle any major changes. These are the top three that we look for. We look for caddisflies, and some people might know them, especially any anglers might know caddisflies. We look for stoneflies, the nymphs, the larvae, and then again, this is for what they look like when they mature, and then the mayflies. And we all know when there's a hatch out, especially of the mayflies, your windshields will be covered with them. Our picnic table in the backyard gets covered with mayflies. Mayflies are a fascinating animal. They live several years in the water. In the aquatic system, their larva is such that once they hatch, they only live for 2 to 3, maybe 4 days at most. So they live the majority, 99.9% of their life is in an aquatic stage. These three are extremely sensitive, in part because of their physiology, in part because of where their gills are. So in this particular Ephemeroptera, or mayfly, you can see that the gills are on its what we call the abdomen. And so this is its breathing mechanism. With the stonefly, they're underneath, depending on the species, but they're, for the most part, they're underneath their arms. When we see these, when we actually get them in a sample, they do push-ups a lot to get water to filter over their gills underneath them. And then the caddisflies, they come in all different shapes and sizes. And so they have different ways of eating, or excuse me, of eating and feeding and breathing is the word I'm looking for. We also have some really beautiful different organisms that are living in these streams and rivers around us. And this is a picture was taken by Connor, who's going to speak in a couple of minutes. of this particular case-making caddisfly. The caddisfly, certain species will use whatever they can find for their casing. That's their shell, basically, where they live. And so some of them are just rocks and sticks and detritus all mushed together. And then there's some others that are just very particular about the material that they're using and how they're actually building their cases. Beautiful. So we find stoneflies. This is a giant stonefly. We quite often find some other insects and bugs out there. This is a predaceous water bug, and I always say to students, watch out for these. Are not ones you don't really want to pick them up. The student was very fortunate; she didn't get bit by it. But hellgrammites and certain types of water beetles are pretty. They can be pretty painful bites that they leave. We are looking at, when we do our bioassessment, a variety of different types of streams and tributaries that feed into the Black River. We go to the higher elevation streams that have a very different substrate. They have cobble and boulder and gravelly, and this provides a lot of great habitat for different species of these bugs. There's usually a lot of very fast moving water over these rocks, so the dissolved oxygen that's in the water is really creates a perfect living condition for some of those really sensitive species with their gills on their abdomen or under their torso. And then the water temperature is also critical as well. So in the higher regions, we'll find faster moving water, quite often a canopy cover that provides for nice cool water, which then also helps to hold the dissolved oxygen. This is down in the village, and I just put this picture in here in part because we have a faculty member for many years, Dick Smythe, who was there with some students. We also have a lot of issues with Japanese knotweed. So while it's a beautiful section of the river, invasive plants are also becoming a tremendous problem, I know, all around the state, and we're seeing it now in Craftsbury. Just another shot from that river. So students are they're sampling in big sections of rivers. They're also looking at tiny little tributaries, little streams that are adding into the larger Black River watershed. And this is where the Black River begins to meander and gets bigger and bigger, and it ebbs and flows in terms of its volume based on the season. The students in the class get prepared to go out into the field using a variety of different tools. The major tool that they use out in the field are these nets that they'll use to actually collect the macroinvertebrates. But they also have with them some small, very informal, or excuse me, simple types of tests for testing, or kits for testing phosphorus and nitrogen. They're also out there measuring stream bank failure. They're looking at the depth of the river, how far the banks are actually collapsing into the river as well. And they're looking at rivers of all different types, or excuse me, of all different sizes, water bodies. And so we have students, this is a site near a farm that were what they thought was going to be a good site for different macroinvertebrates. The family composition that was found there, the families that are associated with this particular area, has high phosphorus and really silty bottoms. So those really sensitive, the mayfly, stonefly, and caddisfly, those dropped out of the sample. And we had other species like diptera and some other crane flies, black fly larvae that don't, that can really be in areas that that they're more pollution tolerant. That's what I'm trying to say. So students are out, because the courses are short, we're out there pretty much rain or shine. We've actually been sampling out in the winter. I do put the students, they're not only in waders, but I put them in life vests too if the rivers are really running. But they're putting in the net to gather the bugs, or washing or just dislodging the bugs off of the rocks. And then they'll be looking very closely to see, what their sample looks like. They spend a long time picking away at the different bugs, collecting them to take them back to our lab so that they can then begin to identify these particular animals that are there. I do need to say that when we did a big study with the National Science Foundation grant at Sterling back 10 years ago, we were using ethanol. So we were taking these organisms and preserving them. But for the class purposes, the students are able to look at them closely. They sort them out by family, and then we are able to bring them back to the river. So by sorting them out, it gives us a better sense of that family composition. So we're putting them, sorting them by what we refer to as orders before we get to family. So the hellgrammite that we see here is a particular species that's semi-tolerant of pollution, but it also needs to have clean water. So when we find hellgrammites, we know that the river might be providing a variety of different types of water quality for it. When the students, after they gather all that information, they input the data. This is just to order, but they're looking specifically at how many of the really sensitive ones, once again, the Ephemeroptera, the mayflies, or the Trichoptera tricatus flies, or the stoneflies, what the percentage of those are in the full order, so, or the full sample. So in this case, we have the blue indicating, I believe that's mayflies, and then with the stoneflies and the caddisflies. So in the samples that we took through Cass Brook, which is a tributary into the Black River, as well as the Black River Village, as well as another higher elevation stream, Seaver Branch, the students were able to see that all three sites, even though the land uses were different, they all still had a good percentage of the three species that are very sensitive. When we take that information just a little bit further, that's when we really get to have a better picture of what's going on. So actually, let me just say here, this is a sample that was taken off of the Cemetery Road. And while they're the Ephemeroptera, the mayflies, the copper stoneflies, they're still there. And even the, excuse me, cat needs to go away. Even the Trichoptera, they're all, those are simple or sensitive species. They're all there, but the higher percentage is in more of the diptera. So we're looking again at crane flies, black flies, and some of these other organisms that can tolerate pollution. That gives us a heads up that something is going on in terms of the water quality. We still have those sensitive species, so we know that there isn't a real dire situation in terms of the water quality. It's just that there's more of the this one particular order, excuse me, and that could easily be the cow input. And this is from a while ago, this data. In this particular sample, the students then took the caddisfly and they started really taking it down to family. Each of the families within the caddisfly order, those particular families are telling another story. So there was a tremendous amount of caddisfly in this one stretch of stream, or stream that's outside of the village on the way to the, where the river really begins to grow. This is by Pete Screen's farm. So there are the sensitive species there, but there's a lot of, you can see here, it says Hydropsychidae, that pink area. They're again, very sensitive species, but that family can handle a certain amount of phosphorus. So the fact that there's such a large percentage in our sample is also another flag that says there's phosphorus present. Where is it coming from? It could be coming from the village and the septic systems in the village. It could be from sedimentation. That would be a flag that would then cause us to want to go back and do some further analysis. This particular stream, this site here is just an upper elevation stream, the Seaver Branch, and this is looking at its caddisflies and, excuse me, they're stoneflies. And the stonefly families are so diverse up in this one little stretch. That gives us a real sense of an understanding that the actual river is really clean and provides all the habitat needs for this diversity, these multiple families within this sensitive species. Okay, I'm going to turn this over to Connor. And Connor, you can drive from here, or I'll keep driving, but you can talk from here.
Okay, great. Thank you, Farley. So again, I'm Connor. I'm a student of Farley's. I've taken almost every one of her classes and a number of independent studies. And right now she's one of my co-advisors on my senior year research project. So I became really fascinated with the idea of bioassessments through taking Farley's Watershed classes. I thought it was amazing that you could use organisms to tell a story about their environment. And so that inspired this project that we worked on together, looking at the seasonality and how does the winter season influence macroinvertebrate communities in the Black River watershed, and how does that affect bioassessments? So with bioassessments, it's really important to have a reference to sort of look at a stream over time, sampling around the same time every year. So you can say, you know, for example, if there was a lot of sensitive species one season, and then the next year at the same time took a sample again, you lost all those sensitive species, that would be indicative of something that's happening in the ecosystem. So we sampled in winter, which is in the Northeast, an out of index period. A lot of organizations will sample in the fall when the flows are relatively low and it's more safe, more or less, to get into streams. So we went out in the wintertime, and these are just some students and friends of mine that helped me, and that Farley and I sort of coerced into getting into the water in the middle of February with us. And Farley, if you go to the next slide, we can talk about what we found. So there are seasonal variations for macrovertebrate assemblages in streams, which makes sense. All these species have different life histories. So in the winter, when one species might be floating around and eating and swimming, another might be buried deep into the sediment, or it might be in an egg stage, something that wouldn't show up in a sample. So in our surveys in the Black River watershed, there was many species that weren't present in the winter that were present in the fall when we would typically do this. So index period matters. When you sample matters, and it's really important if there's an event in the wintertime and you want to look at how the macroinvertebrates respond to that event, you're only going to really be able to compare that to other winter sampling times. You're not going to be able to compare that to the fall. And so other organizations are doing similar studies to sort of get an idea of what macroinvertebrates are up to in seasons when people wouldn't typically sample. And Farley, if you go to the next slide, you'll see that the Vermont Department of Environmental Conservation is doing an out of index period sampling study too. In the fall, or in the summer of 2019, I worked with them on this project, where they're also doing an out of index period sampling. So they were doing it in the summertime as opposed to the wintertime. And so this is with the group at the Watershed Management Division, part of biomonitoring and assessment. And they are on a schedule where they sample streams all across the state of Vermont to do this long-term biomonitoring study. And this can be used to indicate how organisms respond to like massive weather events like Hurricane Irene and things like that. And so for this study, they were looking at, okay, if we want to sample in the summer, what are we going to compare that to? So for this summer, they were going out and collecting reference sort of reference data to compare future data to. And their macroinvertebrate identification is very in-depth. They're very interested in the species of some pretty ubiquitous macroinvertebrates. The picture on the top right is the head capsule of a midge, which is closely related to mosquitoes, and sort of broad scale, they're considered very pollution tolerant, but when you look at them at the species level, some organisms are a little less tolerant, or are a little less tolerant, and some are actually particularly sensitive. So sometimes it's not great to use sort of umbrella sensitive and tolerant for organisms. And when the state is doing really in-depth sampling, they pay particular attention to that. Farley, could you go to the next slide, please? And there's, besides monitoring habitat quality over time, or stream quality over time, biomonitoring also has other applications for conservation of other freshwater species. So this is a chop and drop project that I worked on in this past summer of 2020 with Tim Mountain Conservation Center in New Hampshire, which is out of the Black River watershed, out of Vermont, but there's projects like this going on in Vermont right now. Some chop and drop projects in the Nulhegan Basin to restore brook trout habitat. And the premise of this project, really quickly, is to sort of recreate these old growth forest conditions in streams. So referring to what Farley was talking about earlier, when there was massive deforestation across the northeastern United States, the streams looked much different prior to that. There was on average 4 large trees per every 100 foot of stream, and that's not the case so much anymore. Stories from before indicate that brook trout were much more plentiful before massive clear cutting events. So this project was to positively influence brook trout habitat by dropping trees. And a big part of that was using bioassessments, because macroinvertebrates play a huge role in brook trout life history. They sequester food from plant debris and other microorganisms in streams to create protein to feed brook trout. And so it's really important to monitor them over time and see how macroinvertebrates respond to these massive chop and drops. And Farley, if you go to the next slide, I'll talk about some other organisms that you can use as bioindicators, specifically microorganisms. So this is a fantastic group of fungi that live in streams, and they're fairly ubiquitous. Not a lot of folks know about them, and I just think they're absolutely gorgeous. They play this really interesting role where they're essentially the intermediaries between leaves and other plant debris that fall into streams and the macroinvertebrates that we've been talking about, and then the trout. So these are the primary decomposers of plant litter in streams. And so without these fungi, trout and macroinvertebrates would not be doing so well. And these pictures on the right are some spores of the aquatic fungi that were collected from a stream on Sterling College campus, which feeds right into the Black River. And they have these fantastic shapes as an evolutionary adaptation to life in water. So a typical fungal spore, which is more or less ovate, would have a hard time latching onto substrate in a quick moving stream. But these star-shaped, tetradiate shaped spores are able to latch on to leaves as the water is pushing them by, and propagate and sporulate and break down leaf litter. But they're also indicators of environmental quality. And Farley, if you go to the next slide, there's just some other great pictures of them, and this is the last of it, but they're also indicators of environmental quality. So there's ways that you can go out and sample these organisms to indicate whether a stream is being polluted or whether it's in pristine condition. And so the idea of bioassessments can be applied to a lot of organisms. And this is just another example of that. And that's all I have. So Farley, I'll turn it back to you.
Great.
Thank you.
Yeah, thanks, Connor. And Connor and I have taken given a picture, a very microscopic picture. I want to zoom back out again and talk about what it is that we can do, what we're doing on the landscape. The Craftsbury Conservation Commission has done a lot in terms of trying to better understand native plants, but mostly invasive plants or non-native plants along the river. And the Conservation Commission is doing a tremendous job of trying different methods to eradicate Japanese knotweed. But that said, the Conservation Commission was also really interested in the stream bank stabilization of the Black River as it was coming into the village. So I had shown early on, I showed where the stream bank was failing by our town hall. This is our sand pile for our roads. So it was failing back here. And the town owns a good chunk of this land, this property. We we signed a contract with Natural Resource Conservation Service for a Trees for Streams project. And so we're in a contract for about 10 years, I believe, where we will not mow this property. We'll let it grow back on its own, but we've also planted about 150 trees there. So the idea, once again, is to help stabilize the bank, the stream bank, and so we don't have that failure happening, and thus the sedimentation, which again carries phosphorus, but also has major implications in terms of the fish habitat and the macroinvertebrate habitat. The other project that we've been working on in the upper reaches of the Black River is stream bank stabilization using fascines, which are bundles of willow that once they're actually put into the banks of where there's a lot of stream bank failure happening, they will take hold. And then, as a lot of folks may know, that willow regenerates really quickly. So hopefully in a year or two, we'll actually see some regeneration. This is Australian students working with folks from the U.S. Fish and Wildlife on a parcel of land, just above the village. And so the students worked to actually cut into the bank and to bury those fascines. And again, hopefully at some point soon, we'll be able to see an established natural community of willow to help stabilize that bank, to be really using a biological way to stabilize for restoration of a stream bank, as opposed to riprap or any other type of armoring that might happen that may not work as well. And we would be more than glad to take some questions. I do have this fantastic video that we would love to show that Sterling College produced from the Fish and Wildlife, or the Wildlife Conservation class, about electrofishing. But we have all tried to stream this, and I know Lisa was doing a great job trying, and it's just a big program. We're going to send you the link at the very and when Lisa actually sends out the evaluation for tonight.
Excellent. Thank you so much, Farley. I just really love the way you and Connor talked about it, a story being told through the invertebrates in the stream. I really think that's a great way to look at it. There's so much that our natural world, the story our natural world tells us, and it really is a story that we can enjoy and listen to. So I liked that framing of it. Thank you so much.
Sure.
There's, it's funny, you answered some of my questions at the end, because I'm like, what'd you learn? What are you doing with what you learned? So thank you for that too. I am wondering though, like, have you really seen, or have you graphed, or the students graphed? You said you've been doing this for almost 11 or 15 years.
Yeah.
Have you,
Yeah,
how is the changes been? And I know Connor explained there are seasonal changes, but what are you seeing for if you just graphed the season that you've been doing?
Right. We have the early on data set, I should start off by saying, is a little bit wishy-washy. I mean, some of the students they cut their teeth on the beginning of this class. I mean, this class is a way to learn about the macroinvertebrates. It's when we have students who are doing upper level research like Connor, and I've had some other students in the past who have given us some really good data sets, and that, yes, we can go back to that. So the great thing that we've been seeing is some of the landowners who are actually doing stream bank restoration by planting and preventing some of that that sedimentation getting into the waterways. So as we were showing where the fascines are going in, but even further on up, Pete Johnson on one of the fields he's working on, he did a Trees for Streams project, and we've worked with him to help do some of the willow planting too down by the main part of the Pete's Greens farm as well. So long way of saying that, yeah, I have images from 10, 15 years ago, pictures of showing the land was not wide open, but bit more disturbed, and now it's really beginning to regenerate or to restore into a vegetative state.
Okay, great. Yep. So there's a question back earlier. We talked about ones that were pollution tolerant, ones that were less pollution tolerant in terms of the macro invertebrates. And someone asked, Michael asked, are black flies pollution tolerant?
Yes. Well, this is interesting. Yes, black flies, they are pollution tolerant. Doesn't make them. We said we want them there. Actually, we don't want black flies, but they're a tremendous food source for fish, but also for certain other animals, birds and so forth. But they are definitely, we can find them in some of those more protected or healthier streams. They're just, if the habitat's there for them, not really fast moving water or even stagnant water, they can survive. But we do see them a lot in ditches. So if we're looking at a ditch that may be filled with sediment but has some water in it, that's no longer able to connect to or is connected to a broader water system, those ditches fill up with not just black flies, but scuds or some of the other species, other type of aquatic worms that we'll find. Just this fall, students were working with a farm down in the Black River, down in the village off the cemetery road, and one of the areas where the water's draining off of the through the manure pit, and they were finding quite a bit of aquatic worms. And the farmers down there have been fantastic about working with NRCS to do some stabilization and some restoration, addressing some of the phosphorus loading that's going on there. So finding those aquatic worms definitely is a big flag, as well as black flies.
Interesting. They also wanted to know, are rusty crayfish present in your sampling?
Oh, that I don't know. We haven't come across them. We'll just leave it at that. But no, we do, yeah, periodically. When we electrofish with Judd Kratzer from Fish and Wildlife, we quite often pull up crayfish, but I don't know what species they are.
Okay. Will you do any data analysis with using GIS mapping? Do you do that?
Well, actually, I'm going to turn it to Connor just really quick. We haven't taken our data sets yet and use the GIS mapping with it, but we do have a GIS program, or 2 classes, an intro and an advanced class. And the students do have to do a mapping project. So I was just going to say, Connor, real quick, did you have in your intro class, you'd use GIS to model the movement of water? What was your project?
My project was focused on using GIS data to locate parcels of land around the Black River that are likely to contribute sedimentation. And that was through a combination of looking at the slope of the land and the amount of, or lack thereof, vegetation along the riparian zone. Yeah.
Okay. Yeah.
Oh, Kevin said you had just answered it, but Kevin wrote, in Craftsbury, the Jones farm looks like they put in a type of manure collection basin, and then evidently you answered it. So we're all good there.
Yes. Yeah. They've done a lot of great work. Yeah. Yeah. And
yeah, it's really fun to recognize the good work that's being done.
Yes, exactly. And it was interesting when the students were out on their property this fall. They recognized the farmers could say, oh, this area we really shouldn't have this, the cows moving across this part of the stream. We're going to take care of it. So they're aware, they know, exactly what needs to be done. There's a lot, in a farmer's day, but yeah, to take care of, but they're very aware. It's great.
Brian wants to know if there are any knotweed success stories. And I think he means treatment, not eating of.
So is this from Brian McChesney? No, because Brian McChesney, who's in Craftsbury, he's been worked with us years ago using weed wrenches to take out honeysuckle and other species, invasive plants. The Conservation Commission has a plot right now, an area that's owned by Fish and Wildlife, the land is, and it's a demonstration area. So we are using a variety of different techniques, including the direct application of herbicides, as well as just smothering the sites with plastic. And then another idea that we've been talking about is actually using hardware cloth to try to keep back the shoots after we're cutting on a daily or on an annual basis.
And is anyone better than the other, or how far are we in the research to that? And
well, we're probably a good 5 years into it, but as a lot of people know, knotweed, it takes a long time to get it under control and to see how it's going to respond. It just takes perseverance and diligence. Yeah.
All right. Kevin wants to know, does phosphorus last a long time in the soil? And if so, past heavy usage of phosphorus on the soil will still have an effect then? So if it has a long half-life, that's the way they talk about it.
Right. So phosphorus is naturally occurring in soil. It's naturally bound up in soil. I don't know enough to say how long, if phosphorus is applied to the soil, how long it will remain bound up in it. So I can't answer that question.
There is a question. Are farmers, is there a program, and it might be through NRCS, for them to sow winter rye in the fall for soil retention?
Good question. Yeah, I don't know. It'd be great to hear from NRCS about that.
And we do have NRCS sometimes speaking with us, and may do another Zoom again with Toby Alexander, who is involved with the NRCS program. So stay tuned for that, or sign up for our e-news. Connor, there's what are you doing? This is your senior year. It's been an interesting senior year. Do you have plans for after graduation?
Well, right now I'm really focusing on my senior year research project. I haven't given after graduation much thought yet, but yeah, I would love to keep working with water. I think water quality and the organisms that live in streams are just fascinating, and it doesn't seem like work or studying. It's just something I enjoy. So anything in that direction would be absolutely wonderful.
And if I could just jump in and say that Connor's an example of a Sterling student who has made connections with the state of Vermont, with other nonprofits or organizations, and has done internships and he's building his resume. So along with his undergraduate research, as well as his connection to these groups, is really kind of setting the stage for him when he walks out. I will be hearing from you in the future, I know, Connor, as the expert of fungi in the water or macroinvertebrates.
Yeah, that was really cool. I hadn't known that about fungi. So, it's always something new to learn. So that was very cool. So thank you. Well, we're approaching 8 o'clock and there really aren't any more questions in the chat right now. I'm sure Farley would be happy to answer any questions offline if people are uncomfortable asking them here. You can find her contact information on the Sterling College website, and I'll put it in the email when I send out the video link in case you want to share it or watch this again. There'll be a brief evaluation and then also the link to the video that we couldn't quite share tonight of the electroshocking. So that will all come to you probably Monday, just to let you know. But Farley, thank you so much. And Connor, thank you. Great to hear from you. And it was a great evening. Thank you all very much. Have a good night.
Thank you.
Thank you, Farley.
So, see you, Trevor. Bye.